I would like to begin by thanking the organizers for inviting me to attend the meeting and to give a talk. This photo, taken a number of years ago, shows me holding CPK, space-filling molecular models, of actinomycin D intercalating into what I have called the beta DNA structure, this being a metastable and hyperflexible liquid-like phase that acts as a transition state intermediate in DNA melting. Beta DNA is proposed to arise within entities called Premeltons, these having dynamic structural properties that allow drugs and dyes to intercalate into its structure. Their presence in DNA leads to a unifying conceptual theory to understand much of DNA physical chemistry and molecular biology. In particular, premeltons are predicted to define the 5' and 3' ends of genes in both naked DNA and DNA in active chromatin, this having important repercussions for understanding physical aspects of the initiation, elongation, and termination of RNA synthesis during transcription. For these and other reasons, the model will be of broader interest to the general audience working in these areas. It makes a number of experimental predictions, all readily testable, as will be pointed out in my talk to you this morning. So, let's get started. After our initial studies with actinomycin, we continued to discover a large number of additional crystalline complexes containing planar intercalators complex to a series of self-complementary DNA and RNA-like dinucleoside monophosphates. Their interactions with these nucleic acid fragments are simple, meaning that they exclusively utilize stacking interactions with the base pairs and electrostatic interactions with the sugar phosphate chains to stabilize their structures. One such structure is ethidium ribo-CpG. The complex consists of an intercalated ethidium molecule, shown with dark covalent bonds, and stacked ethidium molecules, shown with light covalent bonds, located above and below the intercalated complex. Sugar phosphate chains demonstrate the mixed sugar puckering pattern C3' endo linked 3' 5' C2' endo, which allow base pairs to separate 6.7 angstroms and to remain twisted relative to one another by about 10 degrees. We refer to this base pair dinucleotide structure as being the highest energy form of the beta structural element pinned by ethidium. This beta structural element has been observed in 15 separate crystallographic determinations. These involve 7 different intercalators, complex to a variety of DNA-like and RNA-like dinucleoside monophosphates. 4 structures are isomorphous and therefore demonstrate a host-guest relationship. The remaining 11 structures crystallize in different lattice environments that contain varying numbers of water molecules. This information readily leads to the ethidium DNA neighbor exclusion binding model shown here. The beta structural element plus ethidium form the asymmetric unit of the helix. A repeated twist of 47.2 degrees and a translation of 9.8 angstroms along the helix axis generates the helical complex shown. It should be noted that intercalation occurs between every other base pair, since binding is restricted to neighboring beta structural elements. This feature explains the magnitude of DNA stretching and unwinding accompanying neighbor exclusion binding. Notice that the stereochemistry connecting neighboring beta structural elements is different. C2' endo linked 3' 5' C3' endo. there is no significant stretching or unwinding in this region. An important prediction of this neighbor exclusion model is that extended microcrystalline domains be present at high drug-to-DNA binding ratios. This prediction has been confirmed by fiber diffraction studies, which indicate a platinum organometallointercalator to form extended microcrystalline domains when it complexes with calf thymus DNA. From the sharpness of the 10.2 and 5.1 angstrom near-meridional reflections, these primarily reflecting the platinum-platinum scattering vectors, the crystalline domain size in these fibers can be estimated to be in the order of several hundred intercalated base pairs. This most easily being understood as arising from a structural phase transition, in which this platinum organometallointercalator, highest energy beta DNA complex, arises as its dominant phase. The beta DNA structure is proposed to be both metastable and hyperflexible and, therefore, to exist in many different energy states. It is bounded on the left by its lowest energy state and on the right by its highest energy state. Steroidal diamines such as iridiamine A stabilize its lowest energy state by partial intercalation, while planar drugs and dyes such as ethidium stabilize its highest energy state by complete intercalation. The lowest energy state is proposed to be a transition state intermediate in the B to A transition, while the highest energy state, being a maximally extended and unwound DNA duplex structure, is proposed to be a transition state intermediate in DNA melting. For comparative purposes, each structure shown contains 20 base pairs. As just mentioned, the lowest energy beta DNA structure has helical parameters intermediate between those of A and B DNA, suggesting it to be a transition state intermediate in the B to A transition. Again, for comparative purposes, each structure shown contains 20 base pairs. Using the technique of linked atom least squares, it has been possible to compute structural intermediates that lie along the minimal energy pathway connecting B with A DNA. This was accomplished by calculating a series of uniform transitions along the polymer, in which the puckering of every other deoxyribose sugar was altered incrementally, and the structures then energy minimized subject to a series of constraints and restraints. In these calculations, physicists will recognize sugar puckers to be the masters, torsional angles defining the sugar phosphate and base sugar conformations, the slaves. In this way, we have discovered the existence of a minimal energy pathway connecting B with A DNA, which passes through its lowest energy beta DNA form. 25 structures have been calculated by this procedure, although for simplification, only 9 have been shown here. Again, for comparative purposes, each structure shown contains 20 base pairs. These calculations were then followed by a least squares procedure, in which adjacent dinucleotide elements from each structure will link together to form the 2 different kinds of premeltons shown here. It is seen that whereas BA or AB premeltons are globally topological, BB or AA premeltons are globally non-topological. This reflecting the presence of a bifurcation, which gives rise to these 2 different types of structural solitons in DNA. BB or AA premeltons form at specific DNA regions to nucleate site-specific DNA melting. They are stationary and, being globally non-topological, are predicted to undergo breather motions that allow drugs and dyes to intercalate into DNA. BA or AB premeltons, on the other hand, are mobile and, being globally topological, act as phase boundaries, transforming B into A DNA during the structural phase transition. They are not predicted to undergo breather motions. More generally, phase boundaries connecting the central beta DNA region with either B or A DNA on either side are referred to by physicists and mathematicians as the kink and the anti-kink, the premelton being an example of a kink-anti-kink bound state. The modulated beta alternation and sugar puckering, in combination with the partial unstacking of alternate base pairs within these kink and anti-kink boundaries, reflects the presence of the Peierls distortion, a spontaneous dimerization known to occur within solitons that arise in other polymers, for example, the polaron in trans polyacetylene, an electronic soliton that gives rise to its superconductive properties. What are breather motions? This animation demonstrates the lowest amplitude breather motion present within a BB or AA premelton, showing the central beta structural element alternating between its lowest and highest energy conformational states. These hinge-like motions are coupled with the concerted movement of the kink and anti-kink boundaries, shown in the boxed regions, on either side. Such boundaries act as energy domain walls, capable of moving in and out with minimal energy dissipation. Isoenergetic breather motions such as these demonstrate the collective effect, an effect well known in many areas of physics. Small movements of atoms in sugar residues within the kink and anti-kink boundaries combine together to give larger movements of atoms in the central beta DNA core region. This collective effect explains how energy is transiently focused into the centers of premeltons to create an open state into which drugs and dyes intercalate. This animation shows how planar intercalators, such as lithium, are able to intercalate into the highest energy form of the beta structural element, while non-planar intercalators, such as iridiamine A and other steroiodiamines, can only partially intercalate into its lowest energy form. What is the relationship between DNA breathing and drug intercalation? Echinomycin is an example of a bifunctional intercalator having two quinoxalin ring systems separated by 10.2 angstroms, connected through amide linkages to a rigid octapeptide chain. The stereochemistry of this naturally occurring DNA-binding antibiotic necessitates both quinoxalin ring systems be able to intercalate simultaneously into neighboring high-energy beta structural elements, and, for this reason, is a valuable probe to understand the detailed stereochemistry of DNA breathing. And how does this porphyrin intercalate into and out of DNA? Intercalators that necessitate the transient rupture of hydrogen bonds connecting base pairs to gain entrance into and exit out of DNA constitute convincing evidence that DNA breathing and drug intercalation are related phenomena. How does DNA melt? This slide shows a schematic illustration of DNA melting, showing how premeltons become meltons with increasing temperature, these being examples of structural solitons in DNA. Premeltons form at the early melting regions in DNA and, at elevated temperatures, serve to nucleate the melting process. At lower temperatures, kink and anti-kink pairs surround small beta DNA core regions. As the temperature rises, these kink-anti-kink pairs move apart, leaving growing beta DNA cores, whose inner regions begin to experience the nonlinear stretching of hydrogen bonds connecting base pairs. Finally, at still higher temperatures, these hydrogen bonds break and single-stranded melted regions appear, separated from regions of B or A DNA by the complex phase boundaries just described. Such composite structures correspond to higher energy structural solitons and are called meltons. Do premeltons exist at the 5' and 3' ends of genes? This study demonstrates the remarkable similarities between the chemical nuclease 1,10-phenanthroline copper 1, an intercalator, and the micrococcale nuclease in their ability to recognize and to cleave hypersensitive sites in a 5,000 base pair circular DNA fragment containing the histone gene cluster from Drosophila. Cleavage patterns exhibited by both agents are amazingly similar, most hypersensitive sites being found at the 5' ends of genes or lying between adjacent genes. What is even more remarkable is the observation in subsequent experiments that many of these same sites nucleate DNA melting when the single-strand specific DNA binding protein of E. coli is added to these same circular DNA molecules made negatively superhelical. The location of these small melted regions has been established using the S1 nuclease in combination with electron microscopy. Important additional information has been provided by studies of the same gene cluster in active chromatin, where the micrococcale nuclease cleaves hypersensitive sites at the 5' ends of genes, while the pancreatic DNase cleaves hypersensitive sites at both the 5' and 3' ends of genes. Taken together, these data indicate the presence of premeltons at the beginning and ends of genes, playing a key role in determining the initiation and the termination of DNA transcription. Their presence immediately suggests an allosteric mechanism that underlies the formation of the RNA polymerase promoter tight binding complex. One can envision the formation of the transcriptionally competent tight binding complex to involve the initial attachment of the polymerase to a premelton located at or near the start of transcription, shown on the left, triggering a cascade of conformational changes in both the polymerase and the DNA, shown in the middle, that lead to the formation of the tight binding complex, shown on the right. The process described above can be considered to be a series of concerted allosteric transitions leading to the progressive union of two molecular species. How might this occur, and what is its underlying energetics? This is best understood as being a protein-DNA structural phase transition, the emergent phase being the RNA polymerase promoter tight binding complex. Complex formation entails a series of stepwise conformational transitions in which energy is transferred from the polymerase to the DNA in the form of small packets, being referred to as an avalanche of kinks by physicists. This is possible, provided the protein begins by being in a high-energy metastable state. It can then spontaneously fall into lower-lying metastable states as DNA melting and tight complex formation ensue. Such an adiabatic process is expected to have little or no change in free energy. The mechanism is reversible. One can imagine the transcriptionally competent tight binding complex shown on the right to undergo a series of concerted allosteric transitions shown in the middle that lead to the final detachment of the polymerase from the premelton shown on the left. Such a mechanism necessarily accompanies the termination of transcription at the 3' ends of genes. The level of negatively superhelical strain energy in DNA provides the bias that determines the direction of this protein-DNA phase transition. Other more active processes can be involved as well. The tight-binding transcriptionally competent complex arises as a result of topological linking. By this I mean intertwining, not from the presence of a large negative free energy change accompanying complex formation. The extremely large apparent binding constant for this complex, therefore, should not be confused with a true equilibrium binding constant, as described by classical thermodynamics. How does actinomycin bind to DNA? It intercalates into its highest energy beta DNA form, found within the boundaries connecting double-stranded B DNA with single-stranded DNA in the transcription complex. This immobilizes, that is, pins, the complex, interfering with the elongation of growing RNA chains. More precisely, how does actinomycin inhibit nucleolar RNA synthesis? Leroy Liu and James Wang have provided a key insight into the nature of DNA supercoiling accompanying transcription that has shed additional light on this question. They have theorized that, in the presence of significant resistance to the rotational motion of the RNA polymerase and its nascent RNA chain around DNA during transcription, the advancing polymerase generates positive superhelicity in the DNA template ahead of it, and negative superhelicity behind it. In nucleolar genes, where there may be as many as 200 RNA polymerases moving down the DNA template while synthesizing growing ribosomal RNA chains, positive and negative superhelical DNA regions between them annihilate one another, causing adjacent transcription complexes to bond together to form trains of transcription complexes, these now moving synchronously along DNA. If this were the case, then the binding by one actinomycin molecule is sufficient to stop the entire transcription train from moving along DNA. I have recently completed 2 books in the area. The first is entitled Premeltons in DNA, a unifying polymer physics concept to understand DNA physical chemistry and molecular biology. The second is entitled Organization of DNA in Chromatin. Rather than bending uniformly along its length, nucleosomal DNA is proposed to consist of multiple segments of B and A DNA held together by kinks when forming its left-handed toroidal superhelical structure. Thank you very much for your attention.